Receiving our brilliant speaker today, and it is my great pleasure to introduce Professor Ludovic Vallier. I'm so sorry if I don't pronounce your name correctly. Professor Ludovic Vallier, one of the leading international experts in the field of the stem cells and liver regenerative biology. So currently, Professor Vallier is the Einstein Professor for Stem Cells and Regenerative Therapies at the Berlin Institute of Health Center for Regenerative Therapies, and also fellow at the Max Planck Institute for Molecular Genetics. Previously, Professor Vallier also led major research programs, including in Wellcome Sanger Institute, and he established also the NIHR Cambridge Human-Induced Pluripotent Stem Cells Core Facility. This facility is one of actually a key European resource for translational stem cell studies. If I'm not mistaken, so its research focus is more into the molecular and cellular mechanisms, including in the human pluripotent stem cell differentiations toward endodermal lineage, especially in the emphasis of liver and pancreas development, organoid and regenerative medicines, including their applications. Based on his impressive CV, that I know that Professor Vallier is currently managing also several important grants, including European Research Council Advanced, EU Nemesis, and Einstein Strategic Professorships. So talking about the metrics, now Professor Vallier has authored more than 150 peer-reviewed publications, more than 27,000 citations with H-index of 70. And his works include many notable publications, notable papers in multiple papers in Nature, Science, Cell Stem Cells, Natural Biotechnology, and so on. And these papers has provided groundbreaking insights into human embryogenesis, modeling, and also regenerative approaches for liver disorders. So this evening, for some colleagues in other part of the world, Professor Vallier is going to provide us informations regarding mechanisms controlling liver degenerations in human chronic liver diseases. So, Professor, the time is yours, and we can start now. Thank you.
Right. Thank you very much for this very nice introduction. And it's really my privilege to be here today and to present the work of my group on mechanism controlling liver regeneration in human chronic liver disease. So I was mentioned on PI at the Berlin Institute of Health, and also a Max Planck Fellow at the Max Planck Institute for Molecular Genetics. And just before I start my talk, I just would like to disclose potential conflict of interest. So now I've had the chance to be founder and shareholder of different biotech companies, and also to be a SAB member of other therapeutic company. And of course, no, I'm not receiving any funding to give the current talk. So of course, now today, I'm going to spend all my talk to talk about our favorite organ, which is the liver. So I don't need to introduce this amazing organ to the audience, because no, I suspect that you are all working on the same organ. But I just would like to remind you that the two main cell types of the liver proper, in terms of developmental biology and origin, are the hepatocyte, which fulfill most of the vital function of the liver, including detoxification, metabolic activity, storage, and production and secretion, and also the cholangiocyte, which are the cells that line up the epithelium of the BCRT, which is here in green, which is a network of conduit that drain the liver from the toxin, and especially bile acid produced by hepatocyte. And so now most of my talk, I'm only going to focus on hepatocyte or cholangiocyte. And cholangiocyte, one of the key cell types I'm going to mention very often are cholangiocyte, not only because of their function in the BCRT, but also because of their potential capacity to transdifferentiate into hepatocyte. And you will see a big part of my talk will focus on this aspect. So again, no, I don't need to introduce what is our motivation to work on the liver. There is a clear unmet clinical need. Liver diseases are one of the rare group of diseases where mortality is still increasing year on year. And those increase in mortality is, of course, due to a lack of treatment, especially for end-stage liver disease, where the only treatment is organ transplantation, which is limited by the lack of organ donor and also the risk of associated with immunosuppressive treatment. I think I also like to say that this increase in mortality and this unmet clinical need around liver disease is also associated to the fact that in fact, the liver as an organ and liver disease have been kind of ignored or understudied over the years due to the association of the liver with alcohol abuse. And that has been resulted in underfunding of research on the liver, which explains in part some of the delay that we may have in terms of the basic understanding of this amazing organ and the associated therapies. Of course, with the rise of muscle D, now this kind of delay or gap is becoming smaller and smaller compared to other organs. So, of course, my lab is also a basic research lab, so we are driven by the need of new therapy, but also we are also interested by the basic understanding of this organ. And the liver is really a special organ because it's unique among all the organs that we have. I like to say that it's the most regenerative organ in the body, which is true. It's the only organ. If you cut 2/3 of the liver, it will regrow. That doesn't happen with your brain, doesn't happen with your bones, doesn't happen with almost any other system. And there's all other aspects that are really interesting with the liver. It's a polyploid organ. It's involved in a long and complex functional maturation during development. It supports other organs, and including the development of the hematopoietic system. So really, that's really our true drive in the lab is to develop new therapy, but also to better understand the mechanism that control liver biology. And to achieve this objective, we have a program based in Berlin, which really focus on 4 activities. We try to understand liver development, so it's purely basic research. What are the mechanisms that control human development, especially the production of hepatocyte and cholangiocyte, and their functional maturation over time. We also try to understand endogenous regeneration of the liver in acute setting and in chronic liver disease. And we also model disease, especially muscle D, where we try to understand what are the mechanisms that lead to the progression of this disease and what are the mechanisms that induce hepatocyte injury. Ultimately, we aim to combine all the knowledge generated by those different activities to develop new cell-based therapy. And for that, what we want to do, in fact, is not only to produce liver cells, especially hepatocyte, but in fact to produce hepatocytes that are resistant to disease. And what we want to do is really to repair the tissue, but also protect the organ against further injury. So to do that, in fact, we only work on human. So our model system is really human. We rarely use mouse model. We start from patient data, either single-cell transcriptomics, spatial transcriptomics, or even genetics. And then we generate a hypothesis that we validate in vitro using a platform based on human pluripotent stem cells or organoid derived from primary tissue. And most of my talk today will really focus on organoid. And then we perform gain and loss of function or molecular analysis that are impossible to perform in vivo in patient. And we discover new mechanism, new pathway that we like to validate in animal model, but we really use animal model as a preclinical model to validate the therapeutic interest of our finding in vitro. So that gives you the general philosophy of my lab and what we're trying to achieve. Really today, what I'm going to do is really focus on endogenous regeneration, especially in the context of chronic liver disease, and introduce a bit more the model that we use on primary organoid. So I told you, we mainly using human pluripotent stem cells or organoids. So I put a slide on human pluripotent stem cells because that's the historic model we have used over the years, and we continue to use this model. But really, we use human pluripotent stem cells for large-scale production of hepatocyte in the context of cell-based therapy. So in Berlin, we have a program which aims to produce billions of hepatocytes from human iPS cells. We have a 3D protocol in bioreactor now that allows us to produce those beautiful aggregates that express homogeneously hepatocyte markers, including albumin, HNF4, that's secret albumin, and at the level of primary hepatocytes, display a key functional activity, including CYP3A4 activity, again at the level of primary hepatocyte, and get be transplanted in mouse model for liver failure. Shown here in all the green cells, in fact, are human hepatocytes in a mouse liver. So I just wanted to put this slide because just to confirm and reassure the audience that we are still working on iPS, we're still aiming to develop cell-based therapy, and we're progressing that. But today, really, what I wanted to do is to describe a bit of a recent work on regeneration and focus on liver organoid derived from primary tissue. So my lab and others have shown that we can derive organoid from almost all the different regions of the liver, including the BCRT and especially from the BCRT. So we can derive cholangiocyte organoid from the gallbladder, the common bile duct, the pancreatic duct, the intrahepatic duct, and all those cholangiocyte organoids can be grown in vitro in 3D and form this beautiful spheroid that can be expanded for 15 or 20 passage. And I will come back on that. More recently, methods have been published showing that you can derive hepatocyte organoids. So that's the first, really, I think the first paper describing the derivation of hepatocyte, the first derivation of hepatocyte organoid was published in May this year. And those organoids are very different from cholangiocyte organoid. They form this also spheroid, but with a thicker layer of cells, and they can adopt this really yellow color, which is probably due to the bilirubin production. So I just wanted to mention those hepatocytes because that's really going also to transform our field in the coming years. But for now, I'm really going to focus on cholangiocyte organoid, which is the model I would like to describe a bit more. So as I told you, cholangiocyte organoid can be derived from all the different regions of the BCRT. The derivation is very easy. What we do is that we either take biopsy, we cut a piece of the BCRT here, for example, it's a gallbladder. We scrap the gallbladder or the common bile duct. We put that, dissociate the biopsy if it's intrahepatic, and we get this population, this kind of population of cells that, after weeks, we start to form, a week to 2 weeks, we start to form organoid that we can grow in matched in 3D with aspondin, with a twin. We don't need wind to grow cholangiocyte organoid in the presence of EGF, inhibitor of TGF-beta. So very conventional method and culture medium for organoid. And this type of organoids can be derived from almost any patient, and we can derive them even from bile acid or brushing. They are very easy to derive. There is no impact of age or sex on the derivation of this organoid. And in my lab now, we probably have derived more than, organoid from more than 200 patients. And so those organoids can be grown, I told you, for 15, 20 passages and attain the normal karyotype. So they're really stable. They express homogeneously BI marker, including CK7 or CK19. And really, they also express key cholangiocyte markers. They are not, and I will come back on this point, totally identical to primary cells. It's still cells that are grown in vitro. And for example, if you see here, they express in top of cholangiocyte marker, cell cycle marker, since those cells are proliferating a lot in vitro, which of course they don't do in vivo. But otherwise, they express functional markers. And for example, in this case, somatostatin receptor, or SBT, which is a drug transporter, are expressed on a polarized way on the cells, again, suggesting functionality. And we've done a bunch of functional tests on the cells, including bile acid transport, hormonal stimuli like secretin or somatostatin, alkaline phosphatase activity or secretion of gamma GT, and the cells grown in vitro display all those functionality at level that are similar, at least when we can measure them like gamma GT, to their individual counterpart. So really, cholangiocyte organoids grown in vitro are cholangiocytes. So what we done what we decided to do when we generated the data was really try to understand if organoid derived from different region of the BCRT could be similar, because we of course, the BCRT is a complex organ, and each region of the BCRT have different function in terms of bile transport or bile accumulation and bile modification. And so what we wanted to know is really if we could keep this functional difference when we derive organoid from different regions of the BCRT. To do that, we took human cadaveric donors, and we biopsied their liver to collect cholangiocytes from the intrahepatic region, the gallbladder, and the common bile duct. And we either derive cholangiocyte organoid from those 3 regions, or perform single cell analysis on the primary tissue. And then after derivation of the organoid, we also perform a single cell RNA-seq on the organoid. And then we decided to compare all those different regions and the different organoids generated from those different regions. First, as expected, the BI3, the different regions of the BI3 have a different transcriptomics profile. They express different markers, and they can be easily distinguished. Here, you have the common bile duct in green, the gallbladder in red, and the intrahepatic in yellow. And those regions express different specific markers. So, for example, the common bile duct will express high level of SOX4. The gallbladder will express a level of SOX17, and the intrahepatic duct will express also a level of SOX4. And so now we could really distinguish different regions based on those markers. But what was important is that the single cell allows also to establish that those different regions still a black and white expression. In fact, you have a gradient of expression from the intrahepatic to the gallbladder region, and suggesting that in fact those markers are not strictly specific for each region, but correspond more to a gradient of microenvironment in the liver. So then we decided to compare those primary tissue to the cholangiocyte organoid that we derived from the same patient. And what we observe is that in fact the cholangiocyte organoids are very homogeneous in terms of expression. In fact, if you derive a cholangiocyte organoid from the gallbladder, the common bile duct, with intrahepatic, they will have a very similar transcriptome. They really resemble to each other. So which means that when you take a cholangiocyte organoid from any region of the BI3, when you grow them in vitro, they lose part of their regional identity. And that's not was a surprise for us, because in the context of the organoid field, there's a lot of papers suggesting that organoids are identical to cells grown in vivo. That's not really the case, at least for cholangiocyte organoid. Clearly, they lose part of this regional identity. But what was interesting is that if you add environmental factor like bile acid on the organoid, then that transcriptome change, and that's intrahepatic cholangiocyte organoid grown in presence of bile acid. And what happens is that those cholangiocyte organoid start to adopt a profile that's closer to their in vivo counterparts. So that's shown here. Here you have organoids that are grown in different concentration of bile acid, and the more you increase the dose, the more they resemble gallbladder cholangiocytes in vivo. So that's means that really, the cholangiocyte sense the environment, and of course, they will adopt a transcriptomic profile in function of their environment. When you grow them in vitro, they lose this information, so they become a generic cholangiocyte. But if you re-add some of those factors, including bile acid, they will re-adopt an identity which is closer to the in vivo system. So that's an important information, as you will see, because that means that in fact cholangiocytes are extremely plastic. They can change of state very easily, and they are very sensitive to their environment. Also, an important point is that despite the fact that I told you that cholangiocyte organoids lose their original identity, they can really reacquire this identity when they are transplanted back in vivo. And to demonstrate that, what we did is to use a mouse model for biliary injury. So in this case, we inject the toxin in the BI3. And then after we injected the gallbladder cholangiocyte organoid, human gallbladder cholangiocyte organoid, in the intrahepatic system, intrahepatic biliary system of those mice. And what you can see here, it's MRCP, where you can see that if you don't transplant cells, in fact, the mice, the BI3 of the mice is completely destroyed, and of course those mice die. But if you inject the cells, you can reconstruct the BI3. And if you go in vivo and use, in this case, we use human gallbladder cholangiocyte organoid that were expressing the red fluorescent protein, the P, you can see that in fact those gallbladder cholangiocytes can recolonize the intrahepatic system and start to express markers specific of the intrahepatic system. So that means that really cholangiocyte organoid can be really reprogrammed after retransplantation and readopt state that correspond to their microenvironment. And that also means that in fact extrahepatic and intrahepatic cholangiocyte organoid are interchangeable, at least for transplantation. We did the same thing in human, okay, and that's what we did is to transplant gallbladder cholangiocyte organoid in human liver that were ex vivo perfused, so not in patients. So in this case, we take cadaveric donor organs that are not good enough to use for transplantation. We put that in a perfusion system that allows to perfuse warm blood in the organ. So that allows us to maintain the organ for 5 to 7 days. And then like that, we can use those organs for research. And in this case, we transplanted gallbladder cholangiocyte organoid in those organs. And what we observe now is that indeed we could, again, transplant human cholangiocyte organoid expressing ARFT in those organs, and they were able to engraft in the BCRT and to express the intrahepatic cholangiocyte marker. And we could also observe that the transplantation increased bile flow and reestablish reestablish the pH reduction that this transplantation was beneficial for the organ. So now this research is further developed and adapted by Fortis and Positive in Cambridge. We're also developing cell-based therapy approach using cholangiocyte organoid based on those data. So what I've just shown you as an introduction to cholangiocyte and cholangiocyte organoid, that cholangiocytes in the BCRT, in the extrahepatic part and the intrahepatic part, are interchangeable. Okay, they are extremely plastic. They can express marker in function of their localization, but they can also change of state very easily in function of their environment. And that means that we can now use, for example, gallbladder cholangiocytes to generate organoid for cell-based therapy application to repair the intrahepatic region of the BCRT. So just to conclude this first part, I think the main message I want to give you is that cholangiocyte can be easily derived from extra and intrahepatic bile duct. That's really important to underline that all the organoids that have been derived so far from the liver have been cholangiocyte organoids. There's only 2 publications that have truly derived hepatocyte organoid. There's a publication from Toshiro Sato that I mentioned earlier, but also a publication by David Hendricks and Hans Klever. And otherwise, all the papers that have been published have been based on cholangiocyte. Those cholangiocyte organoids closely resemble their in vivo counterparts, but they lose their original identity. The microenvironment is very important. And they represent really, those cholangiocyte organoids are really exciting because they really represent a new source of cells for cell-based therapy application. Even if key challenges remain, but as I told you, Fortis and Positive in Cambridge is starting to address all those aspects. And so, based on that, what we then decided to do is try to understand a bit better what was the function of cholangiocyte, but in the context of chronic liver disease. And that's bring me to the second part of my talk, which is going to focus on regeneration. As I told you, the liver is the most regenerative organ in the body. Now, if you delete 2/3 of the liver, as a solid organ, it will regrow. And it will regrow in a way that is not this similar to what lower vertebrates do in terms of limb regeneration and so on. So really, the liver is a remarkable organ. And this process of regeneration is really used almost every day in the clinic, since resection is the forefront therapy, or at least the forefront clinical approach for liver cancer. So in 2 weeks, you can generate a full, at least one kilogram of organ in human, which is quite remarkable. So this process is well known in terms of acute liver injury. So it's well known that, in fact, the mechanism involved in regeneration is hepatocyte proliferation. Okay? The main process really is that when you cut your liver, you induce reproliferation of the hepatocyte that seems to differentiate, re-enter cell cycle, proliferate 1 or 2 cell cycle, and then regrow the organ. Because that, of course, there's a lot of information that's still unknown, but that's the principle that we know from a number of studies. In chronic liver injury, it's a bit different. So by chronic, I mean chronic liver disease, where you have a limited injury, but a constant injury. In this case, we know that hepatocyte proliferation is probably inhibited, and that in fact they can't repair the liver by simple proliferation, or at least it's more complicated. And the mouse model, this process of chronic liver injury have been broadly studied in mouse model. And mouse model, a lot of mechanisms seems to occur. In fact, no, in mouse model, stem cell activation had been observed, dedifferentiation, proliferation also, transdifferentiation, or even repopulation by other cell type. So it's extremely broad what happened in mouse. And in human, no, for a long time, we didn't really know and fully understand what was happening in human. And or if all those mechanisms that were observed in a normal animal model were also happening in human. So to answer this question, what we decided to do almost 5 years ago was to focus on MASLD or non-alcoholic fatty liver disease and try to understand what happening during the propagation of this disease in terms of regeneration. So MASLD starts with the accumulation of lipids in the liver. And in fact, now 25% of the population will have some sort of fatty liver. Luckily, only a small fraction of this population progress in the chronic form, which start with the cell death in the hepatocyte compartment. The cell death induces a pro-inflammatory response that activates stem cells, which then produce extracellular matrix, inducing scarring, which increase cell death in the hepatocyte, more pro-inflammatory response, more stem cell activation, more scarring, ultimately resulting into fibrosis, cirrhosis, and end-stage liver disease or cancer. And so that's this process is relatively well described in human, but really what are the mechanisms that occur during this process, especially in terms of regeneration, are still under debate and relatively unknown. And to address this question, what we decided to do is to develop a single map of the disease progression in human. For that, we collected biopsy from patients that underwent diagnostic for MASLD MASH. And half of the biopsy were used by the pathophysiologist to score the disease. And the other half was used by us to extract nuclei, because we perform single nuclei analysis, to analyze the transcriptome of the liver cells. And using this approach, we are able to collect on 100 patients and from a diversity of state of disease, like a stage of disease. And we were able to collect from LC, from cadaveric donor, from NAFLD early stage, intermediate stage, MASH, late stage cirrhosis, but also end stage, meaning patients that were explanted and receive a transplantation. And what we observed, because we use single nuclei analysis, we were able to isolate nuclei from all the different cell types of the liver, including hepatocyte, which were the most abundant cell type, of course, but also endothelial cells, immune cells, stellate cells, and cholangiocyte. And that's was the first in terms of cellular diversity and also proportion of cells, because usually when you use single cells on a liver sample, you collect many immune cells and kill all the epithelial cells, or in vast majority of epithelial cells. So that's really gave us a good cellular representation of the liver. We are also able to detect disease progression. So for example, here are the hepatocyte, and in purple you can see the end stage hepatocyte, and you can clearly see that hepatocyte now have a strong disease signature, indicating that the progression is affecting their transcriptome. But what was really interesting for us is really this population here, the cholangiocyte and the hepatocyte, and the bridge population that was located between those two populations. Those cells here that I'm pointing out are in fact cells that express both hepatocyte and cholangiocyte marker in different proportions. And in terms of developmental mechanism, they in fact look at cells that are differentiating into each other. So hepatocyte seems to start to express cholangiocyte marker, and cholangiocyte marker that seems to start to express hepatocyte. So that means that we know single cell analysis suggests that we have a biphenotypic population that express both hepatocyte and cholangiocyte marker. Back in vivo and look at if such a population could exist. And so that's LC liver. If you stain hepatocyte and cholangiocyte in LC liver, you see a beautiful cholangiocyte here that express K19 and hepatocyte that express albumin, and they rarely, if never, co-express the same marker. While when you go in disease, of course, what you see it's a very strong fibrotic liver, and you can see what we call a regenerative nodule here, which are the remaining hepatocytes after several years of injury. And in this case, what you see in those regenerative nodule, you can detect the hepatocyte that express both K19, K7, cholangiocyte markers, and albumin, so meaning B phenotypic cells. And we can observe the opposite. Surrounding those regeneration nodules, we can see, in fact, biliary cells that express K19, but also can express hepatocyte marker, including albumin. So that means that really now we were able to detect in vivo those B phenotypic cells that for single cell mapping have detected. And as I told you, for us, based on our understanding of developmental biology, it suggests differentiation between hepatocyte and cholangiocyte, or transdifferentiation. So we went further to try to understand a bit better when this B phenotypic population occurs. So it's progressively increased during disease progression, but it's really, really important in the end stage. That's meaning when the liver is really, really damaged and not functional. And we confirm that in our single cells, but also by immunostaining, showing that we have increased in proportion of B phenotypic cells during disease progression, with the most significant increase in end stage. We then perform pseudotime analysis between cholangiocytes and hepatocytes based on our single cells to try to really understand what was the process that led to this transdifferentiation. And that's allowed us to really detect again what was the population of B phenotypic cells in our single cell map. We then perform RNA velocity, which gave us the direction of transdifferentiation. So we are able to understand if cholangiocytes were transdifferentiating into hepatocyte or vice versa. And what we observe is, in fact, that both situations happen. We have cholangiocyte that seems to transdifferentiate into hepatocyte, and hepatocyte that seems to transdifferentiate into cholangiocyte. So during this progression, it seems that the cells become more plastic and are able to really change of identity during this progression, which of course, can be seen as the process of regeneration. Cholangiocyte is trying to produce more hepatocyte to repair the liver. What is important, we also look at other potential mechanisms. So we look at the presence of fetal progenitor to see if they were a dedifferentiation process, and we look at AFP. And we couldn't really, so we saw an increase of AFP in some patients, but we suspect it's more linked with progression to our cancer. And this population of AFP cells were never linked with the B phenotypic cells we observed either at pseudotime or no UMAP analysis. And the fraction were extremely small. We also look at the potential presence of stem cells, hepatic stem cells, because there's a big debate in the field. And now we try to look at cells that express LGR5 or MKC7, proliferation marker, or TROP2, all those markers being stem cell marker. And in fact, we couldn't find any cells that co-express all those markers, suggesting that stem cell population doesn't really exist in the process. So we do see some, for example, cells that express LGR5, but they are probably coming from Wnt activity and probably not being a specific population of cells that produce hepatocyte or cholangiocytes during this progression. So then now we decided to go a bit deeper in the mechanism that lead to this transdifferentiation. And based on pseudotime, we look at factors that could involve in the production of those B phenotypic cells. And like that, now we identify a number of factors, including, for example, SOX4 and CALAT6 transcription factors that are strongly increased in the B phenotypic population. We also did a number of pathway analysis and observed that the B phenotypic cells are strongly associated with PI3 kinase or, in fact, the insulin pathway in the human. And we validated a few of those markers back into patients, looking at what's happening in terms of expression of those markers in B phenotypic cells. So, for example, SOX4, which was a transcription factor that seemed to be strongly induced in the B phenotypic cells, is not expressed in hepatocyte or cholangiocyte in the healthy liver, or at least not strongly expressed. And when we look at end stage, we can detect this marker in hepatocyte and in cholangiocyte, and we can specifically, in fact, detect high level of expression in SOX4 in B phenotypic cells. The same with CART23. We observe CART23 strongly increase in the B phenotypic population only in end stage disease. So that's mean that based on this data, we were able to detect a number of factors that could explain the mechanism and the process of transdifferentiation. So of course, what we wanted to do next is try to validate those factors at the mechanical level and try to really understand know what could be the molecular mechanism that drive this transdifferentiation during regeneration. And that's where now I'm going to revert back again to cholangiocyte organoid, because now, of course, we have this really great model system that we can go in vitro, and we can use it to simply interrogate key mechanism that we have identified in vivo. So what we did again now is to take cholangiocyte organoid, and this time we only focus on intrahepatic cholangiocyte organoid. But what we did is, and then now we took those cells and then tried different culture conditions to see if we could induce transdifferentiation of those cholangiocyte organoid derived from intrahepatic biliary duct from patients. And what we end up now is, after several trial and error, we likely identify culture conditions that allow us to transdifferentiate cholangiocyte organoid, or at least to induce their transdifferentiation toward the hepatocyte. And so here, that's now cholangiocyte organoids that are transdifferentiated now for 18 days. And what we can observe is that at the end of this process, cholangiocyte organoid contains cells that express albumin. So meaning that now we have cells in those organoids that express hepatocyte marker. What is important is that this protocol doesn't allow the production of fully differentiated hepatocyte. We in fact generate cells that co-express cholangiocyte and hepatocyte markers, so B phenotypic cells. We think the same cells that we can observe in vivo during progression of chronic liver disease. That's shown here by qPCR, where we show the increase of albumin markers, and also the maintenance of K19 expression and cholangiocyte marker. What was also interesting that this model, we were able to see that during this transition process, factors that we have identified in vivo in our single cell analysis, like SOX4, for example, were also induced in those transdifferentiating cells, suggesting that in fact our in vitro model of transdifferentiation is relevant for in vivo process. So now we then apply this method to a diversity of intrahepatic cholangiocyte organoid, and we showed, just to show you that this method works relatively well, that we can derive cholangiocyte organoid from the different lobe of the liver, the right, the left, or the caudate lobe, and that those cholangiocyte organoid will always transdifferentiate and express hepatocyte markers, with some variation in efficacy between patients, age, and so on, but nothing that is when we never observe a case where we couldn't see transdifferentiation. Mechanism that will and validate mechanism that will drive transdifferentiation and with hope regeneration in the human liver. To further analyze and confirm the validity indeed in single cell multiomics on those cholangiocyte organoid transdifferentiating in vitro. And using this approach, now we were able again to confirm the presence of B phenotypic cells, the expression of specific markers that are involved in this transdifferentiation, and to confirm that most of the markers that we have identified in vivo were also induced during this process. And that just show you single cell analysis, showing again the expression of different markers, including albumin, ApoA-B, C2A4, C2C8 in this B phenotypic cell. We also look again to check if we had any stem cells or dedifferentiation, apparition of fetal progenitors, and these kind of things. And again, in vitro, we couldn't really pinpoint stem cell population using this in vitro platform. What we observe again is really a progression of cholangiocyte organoid into cells expressing hepatocyte markers. We couldn't distinguish a specific stem cell population in those cholangiocyte organoid. And we couldn't correlate also this transdifferentiation capacity to the presence of cells that express, for example, LGR5. So really, there is no link seems between the usual cells that are seen as stem cells and the process we see in vitro or in vivo. So clearly, we don't think that there is stem cells in cholangiocyte organoid that will explain this transdifferentiation capacity. So really, our in vitro platform again confirmed that cholangiocytes are very plastic and that this capacity to transdifferentiate into B phenotypic cells, which ultimately, at least in vivo, seems to be able to generate hepatocyte. And then we went a bit further with this platform to demonstrate that some of the pathways we have identified in vitro can also be applied in vivo. And I told you, we identify like PI3 kinase and insulin pathway in vivo based on our pathway analysis on the transdifferentiating cells in patients with chronic liver disease. And what we observe is that if we activate the insulin pathway, the PI3 kinase pathway, we increase the transdifferentiation capacity of cholangiocyte organoid. To block those pathways, we block the transdifferentiation, suggesting that the insulin pathway through the PI3 kinase pathway is important for transdifferentiation of the cholangiocyte into a hepatocyte. We also went a bit further in terms of molecular analysis and tried to identify new factors, or at least factors that at least confirm some of the factors we identified in vivo. And one of these factors was HNF4 gamma. And we were able to detect HNF4 gamma based on the digital footprint analysis on ATAC-seq data sets generated in vitro on cholangiocyte organoid differentiating in vitro. We are able to detect HNF4 gamma on the hepatic gene, especially hepatocyte genes, suggesting that HNF4 gamma, which increased during the transdifferentiation of cells in vivo, could play a role in the transdifferentiation of cholangiocyte. And what we did then is gain and loss of function of these factors in cholangiocyte organoid. What we observe is that the overexpression of HNF4 gamma is sufficient to induce expression of hepatocytes, marker like albumin, ApoB, C2C9, and MMP2 in cholangiocytes, suggesting that really for example, HNF4 gamma could be one of those factors that induce transdifferentiation in vivo during this progression. And now we are validating more of those factors, including SOX4, CALF6, and others to try to really molecular network that allow cholangiocyte to become hepatocyte during chronic liver disease. So just to summarize what we have shown you, and that we'll try to conclude my talk here, is that during this progression, cholangiocytes become more and more plastic. And they undergo a high level of proliferation through what we call Luther reaction, which is part of the process that probably make them more plastic. And this plasticity allow them, but really late in the disease process, to transdifferentiate into hepatocyte, and that could help to repair the liver. But what we also suspect is that this process of transdifferentiation is happening very late in disease, and in fact is also a mark of disease progression and maybe of end-stage disease. And we wonder more and more, in fact, if this regenerative process, in fact, could also go wrong sometime, and in fact be part of more progression toward cancer, and in fact could in fact be an initial step toward tumorigenesis. And that combined with human documentation could lead, in fact, to liver tumors. And that's something that's really interesting to look at. And we are now working more toward this direction in collaboration with Frank Tucker in Charité and Igor Soer. Frank Tucker in the gastroenterology department and Igor Soer in the surgery department. And what we are doing is developing a collection of liver tumor, especially cholangiocarcinoma, where we are collecting those tumors from patients with receiving resection, and we are performing a bunch of deep phenotyping on those tumors, the primary tumor, but also we're trying to derive tumoroid from those tumors and trying to really develop a panel of likely tumoroid that will allow us to validate our hypothesis that maybe plasticity acquisition and transdifferentiation capacity acquired during this progression could be linked to tumorigenesis. And we have just a few first tumoroid derived. For example, that's a liver tumor from a cholangiocarcinoma. And you can see that this tumoroid express K19, but also express high level of HNF4 and albumin, showing that some liver tumor, at least, liver tumoroid will contain a lot of big phenotypic cells, which for us is an indication that maybe again, the mechanism of plasticity we see during this progression could be associated with tumorigenesis. Okay, so to conclude my talk, what I would like to say is that human liver regeneration, at least in chronic liver disease, you have the good, bad, and ugly aspect of regeneration. Clearly, there is the good aspect where chronic injury is going to increase cellular plasticity, especially between hepatocyte and cholangiocyte. And that's going to allow at least some level of repair because the cholangiocyte and the hepatocyte are going to be able to transdifferentiate between each other and repair maybe in part the liver. But then after, of course, this process of plasticity, if associated with somatic mutation, could also lead to tumorigenesis and could lead to further problems. And that's really one of the questions that we're trying to address now. So to conclude my talk, I would like to thank my team, especially all the people that have done all this work. So now the work I've shown on the single cell map and the cholangiocyte organoids transdifferentiation were done mainly by Chris Greben and Vas Ganalakis when they were in my lab. The work now is continued by Marta Cagna. The work on the tumoroid is done by Katia Pusto with Laura Avila and Carla Fro. And all this work will not be possible with the support of our collaborator, especially Irina Moranu, who's done all the bioinformatic analysis I've shown today, and also all our clinician collaborators. And of course, all funding agency, especially the ARC, Patient Foundation, and the BH, we are supporting actively our group to continue this work. I thank you for your attention. I would be very happy to answer any question that you have.